Hi, my name is Felix, and me and my partner Jack worked as a team of 5 for our engineering senior design project, which is also our Keystone Capstone project. And we were supervised by Professor Brianne Canizo. So our project was to design and prototype a mechanical loading bioreactor for real-time imaging of mouse flexor tendon explants. I will try to explain what that means in more detail in the coming slides. So the significance of our project. Connective tissue disorders affect millions of Americans. They cost each individual patient an average of $20,000, and musculoskeletal injuries in total cost about $127 billion annually. Based on these statistics, clearly musculoskeletal injuries and disorders are some of the most pervasive and abused in the United States. And one of the major subclasses of connective tissue disorders are tendon disorders. So you can see what a tendon looks like to the right. It's basically a construction of collagen that connects muscle to bone. And the goal of our project was to better understand and improve our understanding of the structure of tendons and how tendons respond to loads on a cellular level. So we wanted to create a device that allows for a controlled load application to a tendon tissue, while also fitting inside of a high-powered microscope called a confocal microscope that allows for long-term imaging on a cellular level of the tendon while the load is applied. So if you can better understand the structure and function of tendons, this will probably have treatment implications for things like designing artificial tendons and having better connective tissue treatment therapies that have shorter recovery times and allow for a better regaining of motion.
So just a little bit of background about imaging, which is important for our device. We designed this device to be put into a confocal scanning laser microscope. And so basically what this microscope does is you can focus light of a single wavelength on the specimen, which in our case will be the mouse flexor tendon explant. And then you can label biomolecules of interest within the tendon with dyes so that the labeled molecules will emit light of a different wavelength. And that allows clear visualization of those different targets. You can see that really nice fluorescent on black image in the bottom right corner. And so that can be used by our PI for various biomechanics studies with our device in the confocal microscope. And so the particular microscope that we are going to be using is the Olympus FV3000, which you can see at the top right. And so if you open up that hood, there's actually a space constraint of 30 millimeters in height. And so that was a significant consideration that we had to just take into account, was that space constraint. Next slide, please. So the project objectives that we had at the outset were obviously important in our design process. So first we had to design a functioning prototype, and we did that with computer-aided design software, and we had other bioreactors as a reference, and those other bioreactors had similar functions. Not, they had different constraints than us, but they were a good starting point. Then once we had designed the system with software, the other members of our team basically fabricated and assembled all the components. And one thing they had to do was take into account the needs of our PI and choose materials based on those needs. So some of the materials were aluminum, polysulfone, and glass. Glass was important for optical clarity, for viewing in the microscope, and polysulfone, which the majority of our device is made out of, is an easily sterilizable material, which is important. Next slide, please. So the control system was the part of the process that Felix and I were largely responsible for. So on that diagram, in that diagram on the right, you can see an outline of basically the components in the control system that we needed. We basically needed to be able to stretch and compress the tendon, and we also needed to be able to read the force on the tendon, or the load. And so we used LabVIEW, which is a graphical programming language, to program a motor, basically to stretch and compress the tendon. And we needed to be able to control the position of the motor, the velocity or the strain rate at which which is the rate at which the tendon is being stretched and compressed. And we also wanted to be able to move the motor in a sinusoidal fashion, so alternate between tension and compression in the same cycle. And we wanted to be able to track the position data in real time and also export that position data to Excel with timestamps for any kind of later analysis. And then that kind of back half of the circuit basically involves reading the load on the tendon. So for that, we basically set up a load cell, and the load cell essentially changes its electrical output in response to the force it feels. So it's essentially a scale. And so we chose a highly precise load cell that measured load on the tendon when the motor moved. And it basically sent an electrical signal, an analog signal, to a data acquisition system, which then converted the signal to a digital output that the computer could then read, so that we could get a real-time readout of load with timestamps. And then also we needed to save that to Excel as well for later analysis. Next slide, please.
So over here on the left, you can see the image of our completed device. The middle part is an insert that can be removed. And the center part of this insert is a Petri dish, which is glass and allows for imaging from the bottom of this device. These are the grip clamps, which move to stretch the tendon. The tendon is attached in the center, and the motor and load cell are located right here. So this is the grip clamp that moves. The top right is just a picture of some of our code in LabVIEW. And this bottom video is going to show our motor moving in a sinusoidal fashion, as Jack described earlier, forwards and backwards by the same amount. Oh. Okay, so testing. So we tested our device by putting it inside of a confocal microscope, gripping in a tendon, and stretching the tendon while the microscope is imaging. One of the images that we got from this test, you can see on the right. So we dyed the tendon blue so that you could see it more clearly. And each individual dot within this tissue right here is a singular cell. And as it stretches, you can see how each cell responds and moves in response to the load. We checked the position tracking data by first recording it in Excel using our code, and secondly, by recording it using a video, and then using ImageJ to track the position of the motor in real time and comparing those data points with the data points we got from our position tracking using our code. And we found that the R-squared value, which is a statistical measurement method using linear regression, was about 0.99, which means that our motor tracked position very accurately. The photo to the left, which is right here, shows the user interface with our code. So you can see it displays a position graph and a load graph right here. The user can put in which Excel file it would like the position and load to be written to. The user can select the units in which they want to use: millimeters, micrometers, centimeters. Type of movement: relative, meaning relative to where it is now, or absolute, meaning you want to go somewhere regardless of where it is now. The distance, and the speed at which you want it to move, and whether you want the tendon to be in tension or compression, meaning you're pulling it or compressing it. So conclusion and future directions. The load cell data still needs a filter and an amplifier. Unfortunately, the load cell came very late. So we're able to read the signal and record it in Excel, but we need a filter because it's very noisy. And we need an amplifier because the signal is very small. So our data acquisition, our DAC, is not able to read it properly without an amplifier. We also want to add an electrical box because there's a lot of electrical equipment in this device. So we want to be able to store it all in one convenient place. We want to improve the user friendliness of the software interface. But overall, the device was able to successfully stretch and apply a load to a tendon while allowing for imaging inside of a confocal microscope, which was the main goal of the project. Our device allows for multiple loading regimes like cyclic loading that you just saw in the sinusoidal one, or just linear loading, relative and absolute. And it is also able to successfully track the position data collected from the motor, and it's able to track load cell data, even though the load cell data still needs a few modifications. So this video on the right is going to show our device in action stretching a tendon. So basically, you have these grips where the tendon is attached. And then when this grip moves, you can see the tendon, which is in the center right here, gets stretched inside of the microscope, and the microscope can image from the bottom. Thank you. Does anyone have any questions?